Hello and welcome, dear students. Today we are going to discuss about the topic heritability. The main objectives of today's lecture are to know the basics of heritability, to understand about estimating the heritability, to know regression correlation methods of estimation, to know about genomic heritability, to understand the effect of selection and inbreeding on heritability, and finally to know about the conflicts in heritability. Dear students, let's first start with the introduction of the topic. Heritability is a statistic used in the fields of breeding and genetics that estimates the degree of variation in a phenotypic trait or character in a population that is due to genetic variation between individuals in that population. Heritability is estimated by comparing individual phenotypic variation among related individuals in a population by examining the association between individual phenotype and genotype data, or even by modeling summary level data from genome-wide association studies. Heritability is an important concept in quantitative genetics, particularly in selective breeding and behavioral genetics, for instance, twin studies. Behavioral geneticists also conduct heritability analysis based on assumption that genes and environment contribute in a separate additive manner to behavioral traits or characteristics. Heritability is commonly used in twin studies in the field of behavioral genetics. The methodology is based on the fact that identical twins, monozygotic or one-egg twins, share 100% of their genes in common, and non-identical or fraternal twins, that is dizygotic or two-egg twins, are similar to other siblings, that is brothers and sisters, in that they share 50% of their genes in common. The correlation between identical twins is expected to be equal to 1 and that of fraternal twins to be 0.5. In the field of quantitative genetics, the concept of heritability is used to partition observable phenotypic variation between individuals into genetic and environmental components. Furthermore, the heritability concept is subject to misuse when applied to human population differences for traits such as intelligence. For instance, studies have argued that racial differences in measures of intelligence, academic achievement, and crime rates are due to genetic rather than environmental differences. However, other studies have shown that estimates of heritability for such traits within populations do not provide information about genetic differences between the populations. Heritability could increase if genetic variation increases, causing individuals to show more phenotypic variation, like showing different levels of intelligence. On the other hand, heritability might also increase if the environmental variation decreases, causing individuals to show less phenotypic variation, like showing more similar levels of intelligence. Heritability increases when genetics are contributing more variation or because non-genetic factors are contributing less variation. What matters is the relative contribution. Heritability is specific to a particular population in a particular environment. High heritability of a trait or a character consequently does not necessarily mean that the trait or character is not very susceptible to environmental influences. Heritability can also change as a result of change in the environment, migration, inbreeding, or the way in which heritability itself is measured in the population under study. The heritability of a trait should not be interpreted as a measure of the extent to which said trait or character is genetically determined in an individual. The extent of dependence of phenotype or observable trait on environment can also be a function of the genes involved in that. Matters of heritability are complicated because genes can catalyze a phenotype, making its expression almost inevitable in all occurring environments. Individuals with the same genotype can also exhibit different phenotypes through a mechanism called phenotypic plasticity, which makes heritability difficult to measure in some cases. Recent insights in molecular biology have identified changes in transcriptional activity of individual genes associated with environmental change. However, there are large number of genes whose transcription is not affected by the environment. Estimates of heritability use statistical analysis to help to identify the causes of differences between individuals. Since heritability is concerned with variance, it is necessary that an account of the differences between individuals in a population. Heritability can be univariate, examining a single trait, or multivariate, examining the genetic and environmental association between multiple traits at once. This allows a test of the genetic overlap between different phenotypes, for instance, hair color and eye color. A prerequisite for heritability analysis is that there is some population variation to account for. This highlights the fact that heritability cannot take into account the effect of factors which are invariant in the population. Factors may be invariant if they are absent and do not exist in the population, such as no one having access to a particular antibiotic, or because they are omnipresent, like if everyone is drinking coffee. In practice, all human behavioral traits or characters vary, and almost all traits show some heritability. Dear students, let's now discuss key points about heritability. Heritability, the proportion of variation in a particular trait or character that is attributable to genetic factors, is a fundamental parameter in genetics. First introduced by Sewell Wright and Ronald Fisher nearly a century ago, it is key to the response to selection in evolutionary biology and agriculture, and to the prediction of disease risk in medicine. Heritability is not necessarily constant in a population. Change in the method of measurement, environmental change, and the effect of migration, selection, and inbreeding all can alter heritability. The use of high-density genetic marker technologies allows novel estimation methods of heritability. For example, estimation in unpedigree populations and estimation within families free of assumptions about variation between families. The estimation of heritability for new phenotypes, those that can be measured with recently developed technologies, provides knowledge about the nature of individual differences in core biological processes. For example, amounts of gene expression, brain scanning measurements, the length of telomeres on chromosomes, and biochemical compounds measured by mass spectrometry show substantial heritability. Heritabilities are often surprisingly large, and at present there is no consensus theory to explain why heritabilities have the values they do. Fortunately, the incredible pace of gene phenotype discoveries in many species will allow new insight to these questions in the near future. Dear students, let's now discuss about assumptions in heritability. Estimates of the total heritability of human traits assume the absence of epistasis, which has been called the assumption of additivity, Although some researchers have cited some estimates in support of the existence of missing heritability unaccounted for by known genetic loci or position, the assumption of additivity may render these estimates invalid. There is also some empirical evidence that the additivity assumption is frequently violated in behavior genetic studies of adolescent intelligence and academic achievement. Dear students, let's now know about estimating heritability. Heritability may be estimated from the similarities observed in subjects varying in their level of genetic or environmental similarity. The statistical analysis required to estimate the genetic and environmental components of variance depend on the sample characteristics. Briefly, better estimates are obtained using data from individuals with widely varying levels of genetic relationship, such as twins, siblings, parents, and offsprings, rather than from more distantly related and therefore less similar subjects. The standard error for heritability estimates is improved with large sample sizes. In non-human populations, it is often possible to collect information in a controlled way. For example, among farm animals, it is easy to arrange for a bull to produce offspring from a large number of cows and to control environments. Such experimental control is generally not possible when gathering human data relying on naturally occurring relationships and environments. So heritability expressed as H2 is equal to Vg oblique Vp, where H is the heritability estimate, Vg the variation in genotype, and Vp the variation in phenotype. Heritability estimates range in value from 0 to 1. If H or heritability is 1, then all variation in a population is due to differences or variation between genotypes, that is, there is no environmentally caused variation. If H is equal to 0, there is no genetic variation. In this case, all variation in the population comes from differences in the environmental experienced by individuals. In classical quantitative genetics, there were two schools of thought regarding estimation of heritability. One school of thought was developed by Sewell Wright at the University of Chicago. It is based on the analysis of correlations and by extension regression. Path analysis was developed by Sewell Wright as a way of estimating heritability. Dear students, the second thought was originally developed by R.A. Fisher. It is based on the analysis of variance of breeding studies using the intra-class correlation of relatives. Various methods of estimating components of variance and hence heritability from ANOVA are used in these analyses. Today, heritability can be estimated from general pedigree using linear mixed models and from genomic related estimated from genetic markers. Studies of human heritability often utilize adoption study designs, often with identical twins who have been separated early in life and raised in different environments. Such individuals have identical genotypes and can be used to separate the effects of genotype and environment. A limit of this design is the common prenatal environment and the relatively low numbers of twins reared apart. A second and more common design is the twin study, in which the similarity of identical and fraternal twins is used to estimate heritability. These studies can be limited by the fact that identical twins are not completely genetically identical, potentially resulting in an underestimation of heritability. In observational studies, or because of evocative effects where a genome evokes environments by its effect on them, gene-environment correlation may covary. Depending on the methods used to estimate heritability, correlations between genetic factors and shared or non-shared environments may or may not be confounded with heritability. Dear students, let's talk about the regression correlation methods of estimation. The first school of estimation uses regression and correlation to estimate heritability. A basic approach to heritability can be taken using comparison of close relatives, parent-offspring regression, sibling comparison, twin studies. Heritability of traits in humans is most frequently estimated by comparing resemblances between twins. The advantage of twin studies is that the total variance can be split up into genetic, shared, or common environmental and unique environmental components, enabling an accurate estimation of heritability. Dear students, let's now discuss about the analysis of variance methods of estimation. The second set of methods of estimation of heritability involves ANOVA and estimation of variance components intra-class correlations. The use of ANOVA to calculate heritability often fails to account for the presence of gene-environment interactions, because ANOVA has a much lower statistical power for testing for interaction effects than for direct effects. Dear students, let us discuss about the third, that is the linear mixed models. A wide variety of approaches using linear mixed models have been reported in literature, where these methods phenotypic variance is partitioned into genetic, environmental, and experimental design variances to estimate heritability. Environmental variance can be explicitly modeled by studying individuals across a broad range of environments, although inference of genetic variance from phenotypic and environmental variance may lead to underestimation of heritability due to the challenge of capturing the full range of environmental influence affecting a trait or character. Other methods for calculating heritability use data from genome-wide association studies to estimate the influence on a trait or character by genetic factors, which is reflected by the rate and influence of putatively associated genetic loci, usually single nucleotide polymorphisms or SNPs on the trait. This can lead to underestimation of heritability. This discrepancy is referred to as missing heritability and reflects the challenge of accurately modeling both genetic and environmental variance in heritability models. When a large complex pedigree type of data is available, heritability and other quantitative genetic parameters can be estimated by restricted maximum likelihood, that is REML, or Bayesian methods. The raw data will usually have 3 or more data points for each individual, a code for the site, a code for the dam, and one or several trait values. Different trait values may be for different traits or characters or for different time points of measurement. The currently popular methodology relies on high pedigree of certainty over the identities of the sire and dam. It is not common to treat the sire identity probabilistically. This is not usually a problem since the methodology is rarely applied to wild populations, although it has been used for several wild ungulates and bird populations. And sires are invariably known with a very high degree of certainty in breeding programs. There are also algorithms that account for uncertain paternity. Dear students, let's now talk about the genomic heritability. When genome-wide genotype data and phenotypes from large population samples are available, one can estimate the relationship between individuals based on their genotype and use a linear mixed model to estimate the variance explained by the genetic markers. This gives a genomic heritability estimate based on the variance captured by common genetic variants. There are multiple methods that make different adjustments for allele frequency and linkage disequilibrium. Particularly, the method called high definition likelihood, that is HDL, can estimate genomic heritability using only genome-wide association studies, summary statistics, making it easier to incorporate large sample size available in various genome-wide association studies, that is meta-analysis. Dear students, let's now understand the effect of selection and inbreeding on heritability. Quantitative genetic theory predicts that selection and or inbreeding will alter additive genetic variance and as a consequence will also alter heritability. With truncation, selection used in animal and plant breeding, or with stabilizing selection in natural populations, the variance of the parents between family variance in their offsprings is reduced below that of the parental generation as whole. The level of this reduction depends on the intensity of selection and on heritability. Under the infinitesimal model of a large number of loci or position that are each of the small effect contributing to genetic variance, changes in allele frequencies at each locus are infinitesimally small, yet genetic variance is reduced. This change in genetic variance resulting from selection, which is due to gametic disequilibrium, was explained by Bulmer and is commonly known as Bulmer effect. However, if selection stops, then the genetic and phenotypic variances and heritability return to their pre-selection values after a few generations, but the change in the mean is permanent. The results from this limiting model of an infinite number of loci imply that permanent change to the genetic variance and heritability as a result of selection are likely to be small when the number of loci contributing to the genetic variance is finite but large. Individual loci of large effect can, however, be fixed by selection with a consequent reduction in variance. Genetic variance is also expected to be reduced by inbreeding. The rate of inbreeding is a function of the number of individuals who are parents to the next generation and of the variation in family size, which can be expressed as the effective population size. If a parent is inbred, then a proportion of its genome is IBD from a common ancestor, so that the variance of the genic values of the gametes it produces is less than if it were not inbred. The variance within families is therefore reduced by inbreeding. The reduction is linear if the gene effects are additive. Furthermore, inbreeding is higher in a population that is undergoing selection compared with an unselected population of the same number of breeding parents. This is because the best individuals will tend to come from the same families and be more related than when parents are randomly selected. Continued inbreeding is expected to reduce genetic variance slowly but relentlessly, eventually depleting all genetic variances as the population becomes completely inbred unless new genetic variants are introduced. For example, by migration or mutation. Certainly, extreme selection intensity and close inbreeding can result in total loss of genetic variance as seen in inbred lines. These predictions of quantitative genetic theory have been tested in model species and have generally been upheld, although with considerable variation between replication lines for morphological traits in mice, Drosophila melanogaster or fruit fly, and Tribolium castaneum. For fitness traits in which non-additive variance is important, genetic variance has been shown to increase as a result of inbreeding. theory for change in non-additive genetic variation as a result of selection and inbreeding has been proposed, but is difficult to verify empirically even in species in which mating designs can be planned in an attempt to disentangle estimates of higher order non-additive variance components. Moreover, environmental variance has been observed to increase as a result of selection and inbreeding. Dear students, let's now discuss about response of heritability to selection. In selective breeding of plants and animals, the expected response to selection of a trait with known narrow sense heritability can be estimated using the breeder's equation. In this equation, the response to selection R is defined as the realized average difference between the parent generation and the next generation, and the selection differential S is defined as the average difference between the parent generation and the selected parents. Observing the response to selection in an artificial selection experiment will allow calculation of realized heritability. Dear students, let's now talk about the conflicts in heritability. Heritability estimates are inherently limited because they do not convey any information regarding whether genes or environment plays a larger role in the development of the trait or character under study. For this reason, David Murray and David Schenck described the term heritability in the context of behavior genetics as one of the most misleading in the history of science, and argue that it has no value except in very rare cases. When studying complex human traits, it is impossible to use heritability analysis to determine the relative contributions of genes and environment. As such, traits result from multiple causes interacting. In particular, Feldman and Leonotin emphasize that heritability is itself a function of environmental variation. However, some researchers argue that it is possible to disentangle the two. The controversy over heritability estimates is largely via their basis in twin studies. The scarce success of molecular genetic studies to corroborate such population genetic studies conclusions is the missing heritability problem. Eric Tunkiemer has argued that newer molecular methods have vindicated the conventional interpretation of twin studies, although it remains mostly unclear how to explain the relations between genes and behaviors. According to Tunkiemer, both genes and environment are heritable. Genetic contribution varies by environment, and a focus on heritability distracts from other important factors. Overall, however, heritability is a concept widely applicable. Dear students, let's now summarize the lecture of heritability. Heritability is an important parameter in genetics. It allows a comparison of the relative importance of genetic and non-genetic factors across species, traits, and identified environments, and is a key parameter for predicting the response to artificial and natural selection. However, as with all other parameters, it is limited. For example, because heritability is a ratio of variances, it does not tell us about the magnitude of each component. It refers to a particular population and environment and not their differences. Furthermore, because it is a composite measure, it tells us nothing about the actions and interactions of individual genes. Despite continuous misunderstandings and controversies over its use and application, heritability remains key to the response to selection in evolutionary biology and agriculture, and to the prediction of disease risk in medicine. Recent reports of substantial heritability for gene expression and new estimation methods using marker data highlight the relevance of heritability in the genomics era. Heritability amount of phenotypic, that is observable trait variation in a population that is attributable to individual genetic differences. Therefore, heritability allows a comparison of the relative importance of genes and environment to the variation of traits within and across populations. Dear students, it was all about today's lecture regarding heritability. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.